Dear students, after studying this module, you shall be able to know about supramolecular catalytic processes, learn what are host-guest interactions and types of receptor molecules, analyze what are biological mimics, and understand about the concept of co-catalysis. Now, let us introduce this topic. In this module, we primarily focus on supramolecular chemical reactivity, predominantly in accelerating or understanding chemical reactions. There are close parallels between artificial, abiotic supramolecular reactivity and biochemistry. For example, in the study of enzymes and nature's catalysts, synthetic catalysts can both model natural ones and allow the design of new different kinds of reactions. Supramolecular catalysis lies somewhere between chemical catalysis, that is transition metal, and organocatalysis, and biology. Supramolecular catalytic reactions involve binding of a well-defined substrate, that is a reactant, to the receptor, that is a catalyst, and the catalytic process completes in 3 steps. The first step being the selective binding of reactants based on their recognition by the receptor that may bear reactive groups. Second step is the transformation of the bound species. Third is the release of the products and regeneration of the catalyst. The first 2 steps, which involve binding and transformation in this catalytic processes, are the important steps because both steps take part in molecular recognition of the productive substrate and require the correct molecular information in the reactive receptor. Compared to molecular catalysis, a binding step is involved that selects the substrate and precedes the reaction itself. Now, as you can see in this figure, we have shown you the schematic representation of the supramolecular catalysis process. Supramolecular catalysis obeys the general rule stating that catalytic action consists in stabilization of a transition state of the reaction and in the rapid release of the product, which is shown to you in the figure. In a reaction which is catalyzed by the formation of a supramolecular transition state complex, substrates are inserted into either an active site of an enzyme, a host cavity, a layer, a micelle, a vesicle or micropores, bringing their functional groups into a close contact for a relatively long time. In addition to this, the incorporation can stabilize favorable conformations of the substrates. Functional groups in the active site or cavity, such as hydrogen bonding donor or acceptor groups, can additionally exert true catalytic action also. Supramolecular catalysis may be divided according to the type of the aggregates which are involved, since it can be based on a catalytic action of a macrocyclic host, microemulsions, micelles or vesicles, or on the catalytic activity of the mesoporous materials. Now, you can see how the transition state plots for the supramolecular catalysis process is been there, and you can see there how we have drawn them in this figure. Now, students, having introduced to you the topic of supramolecular catalysis, we shall now concentrate our attention to the catalysis by reactive macrocyclic cation receptor molecules. The most important example for catalysis by reactive macrocyclic cation receptors is the deacylation of O-acetyl hydroxylamine in presence of the macrocyclic polyether bearing 4 carbonyl groups derived from tartaric acid. The ability of 18O6 macrocyclic polyethers to bind primary ammonium ions opens the possibility to induce chemical transformation on such substrates. Activation and orientation by binding was observed for the hydrolysis of O-acetyl hydroxylamine. which forms such a stable complex with the macrocyclic tetracarboxylate receptor that it remains protonated and bound even at a neutral pH despite the low pKa value of 2.15 of the free species. As a consequence of this, its hydrolysis is accelerated and exclusively gives acetate and hydroxylamine. Whereas in the presence of potassium ions, which displace the substrate, it yields acetyl hydroxamic acid also, that is amount of 50%. Thus, strong binding may be sufficient for markedly accelerating a reaction and affecting its course, a result that also bears on enzyme catalyzed reactions. Chemical transformations may be induced by a reaction between bound substrate and functional groups borne by the macrocyclic receptor unit. Now you can see here in this figure where we are showing you a scheme of binding of a primary ammonium group by the NH3+ to the cavity of 18O6 macrocyclic polyether to give you the desired molecule. Ester cleavage processes have been most frequently investigated in enzyme modeling studies. Macrocyclic polyethers fitted with side chains bearing thiol groups cleave activated esters with marked rate enhancements and chiral discrimination between optically active substrate. The tetra L-cystenyl derivative of macrocycle 18O6 polyether binds paranitrophenyl, that is PNP esters of amino acids and peptides, and reacts with the bound species, thus releasing p-nitrophenol as shown in the figure. The reaction displays many properties. First is the substrate selectivity, second is the marked rate enhancement in favor of dipeptide ester substrate, third is the inhibition by complexible metal cations that displace the bound substrate. Fourth is the high chiral recognition between enantiomeric dipeptide esters, and fifth is the slow but definite catalytic turnover. Now this is shown here in this figure where you can see the schematic representation of the complex of the receptor 18O6 with the dipeptide substrate, glycyl glycine paranitrophenyl ester salt. A well understood example as mimics for transacylases has also been addressed by Cram, who won the Nobel Prize for supramolecular structures, who also used chiral corands as shown in the next figure, bearing thiolate nucleophiles situated above and below the plane of the macrocycle. Corands of this type can bind primary ammonium cations via charge assisted NHO hydrogen bonds. The host is complementary to the organic cation because of the matching of the threefold symmetry of the NH3+ group with the pseudo six-fold arrangement of the corand binding sites. In case of chiral guests, chiral recognition is observed as a consequence of steric interactions between the substituents on the naphthenyl groups, that is CH2SH in corand, and the substituents on the guest. Guest binding was accompanied by transacylation, in which an acyl group is transferred from guest to host thiol group as a result of the nucleophilic attack by the thiol. In each case, the resolved S host was used, and it was found that in the intermediate, the stabilization of the S host L amino ester pair was significantly greater than the SD distereoisomer. The system thus displays kinetic selectivity in the sense that the substrates of L-handedless react more quickly than those with the D-chirality, because the reaction transition state is more stable for the SL pair. This kinetic selectivity depends strongly on the nature of the amino acid derivative, which determines the identity of the R group on the substrate. for small amino esters such as alanine derivatives, where R is the methyl, the ratio of KSL to KRL is 1. That is, it means no selectivity. The selectivity, however, increases rapidly with the bulk of the R, that is R as it gets branched. However, the rate selectivity factors of 6.4 for R is equal to Me2CH, that is the secondary group, that Me2CH CH2, and 8.2 for R is equal to C6H5CH2, that is the benzyl group, and 9.4 for the tertiary aliphatic carbon group. Now you can see here the key features of the chiral Courant transacylase mimics, and also in the next figure you can see the scheme of transacylation step carried out by the above described Courant, where AR is equal to para-nitro benzyl group. So, hydrogen transfer has been induced with the macrocyclic receptors bearing 1,4 dihydropyridyl groups. Bound pyridinium substrates are reduced by hydrogen transfer from DHP side chains within the supramolecular species. The first order intracomplex reaction is inhibited and becomes bimolecular on displacement of the bound substrate by complexable cations. Reactions with carbonyl or sulfonium substrates have been performed with other DHP containing macrocycles, such as shown in this figure. The earlier one I talked to you about was the bound pyridinium substrates are reduced by hydrogen transfer from DHP side chains with the supramolecular species. Here it is shown to you as structure number 1, and the structure number 2 depicts the reactions with carbonyl or sulfonium substrates have been performed with other DHP containing macrocycles. Now this is shown as structure number 2 in this case. Under the topic of catalysis by reactive anion substrate molecules or receptor molecules, generally the anion hosts also obey the same rules that govern the magnitude of binding constants and host selectivity in cation hosts, primarily based on pre-organization, complementarity, solvation and size and shape effects. Their application is made much more difficult because of some of the intrinsic properties of anions, and these are: first is, the anions are relatively large and therefore they require receptors of considerably greater size than that of cations. For example, one of the smallest anion known is the F- anion is comparable to the ionic radius of the potassium cation, that is 1.33 angstrom versus the 1.38 angstrom. Even simple inorganic anions occur in a range of shapes and geometries. For example, spherical halides, linear SCN, that is thiocyanate, and the azide ions, planar nitrate anions and PTCl4 2-, that is the tetrachloroplatinum salt, and the tetrahedral phosphate and the sulfate anions, octahedral hexafluorophosphate ions, that is the PF6-, and the FeCN6 3-, as well as more complicated examples as in the case of biologically important oligophosphate anions. In comparison to cations of similar size, anions have high free energies of solvation, and hence anion hosts must compete more effectively with the surrounding medium. For example, the delta G of hydration of fluoride anion is equal to -465 kilojoules per mole. However, the delta G of hydration of potassium cation is -295 kilojoules per mole. Many anions, they exist only in a relatively narrow pH window, which can cause problems especially in the case of receptors based upon polyammonium salts, where the host may not be fully protonated in the pH range in which the anion is present in the desired form. Another factor is that anions are usually coordinatively saturated, and therefore they bind only via weak forces such as hydrogen bonding and van der Waals interaction, although they can form the dative bonds too. The development of anion coordination chemistry and anion receptor molecules has made it possible to perform molecular catalysis on anionic substrates of chemical and biochemical interest, such as adenosine triphosphate. ATP hydrolysis was found to be catalyzed by a number of protonated macrocyclic polyamines. In particular, 24N6O2 is shown in the figure, strongly binds ATP and markedly accelerates its hydrolysis to ADP and inorganic phosphate over a wide range of pH values. The reaction presents first order kinetics and is catalytic with turnover. It proceeds via initial formation of a complex between ATP and the protonated form, followed by an intra-complex reaction which may involve a combination of acid, electrostatic and nucleophilic catalysis. The structure from the figure you can see, it represents one possible binding mode of the ATP complex and indicates how cleavage of the terminal phosphoryl groups might take place. A transient intermediate identified as phosphoramidate is formed by phosphorylation of the macrocycle by ATP and is subsequently hydrolyzed. Studies with analogues of ATP, they also indicate that the mechanism was dissociative in character within a pre-associative scheme resulting from the receptor substrate binding. In this process, the catalyst presents prototypical ATPase activity. That is, it means that it behaves as a proto-ATPase. Students, that in this figure, it is representative of the structure which represents one possible binding mode of the ATP3 complex and indicates how cleavage of the terminal phosphoryl groups might take place. A transient intermediate identified also as phosphoramidate is formed by phosphorylation of the macrocycle by ATP and is subsequently hydrolyzed. Multiple recognition and catalysis in ATP hydrolysis with increased ATP by ADP or ADP you can say selectively has been achieved with a multifunctional anion receptor containing a macrocyclic polyamine as anion binding site, an acridine group as the stacking site and a catalytic site for hydrolysis. This is shown as a structure. Phosphoryl transfer is accelerated by other types of hydrogen bonding receptors also. Now in this figure you can see the multiple recognition sites and catalysis in ATP hydrolysis using the molecule. Now let us come down to the discussion of catalysis with cyclophane type receptors. After the macrocyclic type of receptors discussion, the anion type receptor discussion, we will now discuss the cyclophane type receptors. The term cyclophane literally means any organic molecule containing a bridged aromatic ring. By definition, cyclophane hosts, example calixarenes and resorsaccharines, must contain at least one macrocyclic ring and they must achieve closure by some means of curvature. Cyclophane hosts commonly bind both neutral molecules and organic cations, and there is sometimes even more ambiguity as to whether a guest is protonated by, for example, a hydrogen bond, acid host or vice versa, resulting in charge assisted binding. Cyclophanes may or may not contain a molecular cavity large enough to fit host guest species, but in some cases cavities are unnecessary to achieve high binding affinity as long as binding sites exhibiting stereoelectronic, that is steric and electronic complementary to the guest are properly positioned, that is pre-organized on the surface of the host. Thus, cyclophanes can exhibit capsular or nesting type of binding or even apolar surface interactions, often leading to aggregation. Cavities are frequently encountered. However, because of the entropic and enthalpic gains associated with the spherical or three-dimensional encapsulation of the guest by a host, with the convergent binding sites. A number of studies have made use of functionalized cyclophanes for developing supramolecular catalysis and enzyme models. Their catalytic behavior is based on the implementation of electrostatic, hydrophobic and metal coordination features for effecting various reactions in the aqueous media. Hydrophobic species bearing hydrocarbon chains present vitamin B12 or vitamin B6 type activity. Such systems lend themselves to the inclusion in membrane or micellar media. Thus, they provide an important link with the catalysis in more or less organized media such as membranes, vesicles, micelles, polymers. Water-soluble cyclophanes showing, for example, transaminase, acetyl transfer, pyruvate oxidase or nucleophilic substitution activity have been described here. Cyclophane catalysts offer a rich playground for developing novel reactions and enzyme models in view of the variety of structural types, the large cavities they contain and the possibility to attach several functional groups of various nature, of the varied nature. Now we shall focus our attention to the next topic, which is the supramolecular metallocatalysis. Supramolecular metallocatalysis consists in principle of the combination of a recognition subunit such as a macrocycle, a cyclodextrine, a cyclophane, etc. that selects the substrates and of a metal ion bound to another subunit, that is the reactive site. Complexed metal ions presenting free coordination positions may present a variety of substrate activation and functionalization properties. Heterotopic co-receptors such as the one shown in structure bind simultaneously as substrate and a metal ion, bringing them into close proximity, thus potentially allowing reaction between the two reactants or the substrates. Approaches towards the development of artificial metalloenzymes have also been made based on cyclodextrines or macrocycles and involving various metal ions, such as some metals and ions are like zinc 2+, copper 2+, cobalt 3+, for facilitating hydrolysis, epoxidation, hydrogen transfer, etc. Metalloporphyrins have been used for epoxidation and hydroxylation, and a phosphine rhodium complex for isomerization and hydrogenation. Now, another example is the cytochrome P450 model systems. They are represented by a porphyrin bridge cyclophane, macrobicyclic transition metal cyclidines or beta cyclodextrine linked porphyrin complexes that may bind substrates and perform oxygenation reactions on them. A cyclodextrine connected to a coenzyme B12 unit forms a potential enzyme coenzyme mimic. Recognition directed specific DNA cleavage reagents also make use of the reactivity features of various complex metallic sites. The remarkable facilitation of the amide hydrolysis by dinuclear copper complexes and related processes may open routes towards metallocleavage of proteins too. Selective metalloprocesses, in particular asymmetric reactions utilizing external chiral ligands such as hydrogenation, epoxidation, hydroxylation, are of great value for organic synthesis and are being actively investigated also. Supramolecular metallocatalysis, by combining a substrate recognition unit with a catalytic metallic site, offer powerful entries to catalysts presenting shape, region and stereoselectivity. Now, if you have a look at this figure, they are the heterotopic co-receptors for the supramolecular catalysis. Now, the next topic and a very important one is the cocatalysis, that is the catalysis of the synthetic reactions. A further step lies in the design of systems capable of inducing bond formation rather than bond cleavage, thus affecting synthetic reactions as compared to degradative ones. To this end, the presence of several binding and reactive groups is essential. Such is the case of co-receptor molecules, in which subunits may cooperate for substrate binding and transformation reactions. They should be able to perform co-catalysis by bringing together substrates and cofactors and mediating reactions between them within the supramolecular structures. Now this is shown to you here, the schematic illustration of co-catalysis process involving group transfer and litigation reactions. Notice students that in this figure we show you the schematic illustration of co-catalysis processes, that is the group transfer and ligation reactions occurring within the supramolecular complex formed by binding of the substrates of the two macrocyclic subunits of a macrotricyclic co-receptor molecule. Also in the next figure you will realize the process of co-catalysis is being displayed here, that is showing the pyrophosphate synthesis by phosphoryl transfer mediated by macrocycle via the phosphorylated intermediate. Processes of this type have been realized in supramolecular phosphorylation reactions. Indeed, the same 24N6O2 macrocycle as that already used in the study of ATP hydrolysis, as we discussed earlier in this module, was also found to mediate the synthesis of pyrophosphate from acetyl phosphate. Substrate consumption was accelerated and catalytic with turnover steps. That is, the first step is the substrate ACP binding by the protonated molecular catalyst. Second is the phosphorylation step within the supramolecular complex, giving the phosphorylated intermediate PN. Third step is the binding of substrate, that is HPO4 2-, which is the P. Fourth step is the phosphoryl transfer from PN structure to the P structure with formation of pyrophosphate. Release of the product and of the free catalyst for a new cycle, that is release of the product for a new cycle is necessary, and all these steps are also necessary for the new cycle to begin. The structure PP is also formed in the hydrolysis of ATP in the presence of divalent metal ions. The fact that structure is ditopic co-receptor containing two diethylene triamine subunits is of special significance for both PN structures and PP formation. These subunits may cooperate in binding the ACP and activating it for phosphoryl transfer via the ammonium sites, in providing an unprotonated nitrogen site for the structure PN and its formation, and in mediating phosphoryl transfer from PN81 to P. Thus, structure would combine electrostatic and nucleophilic catalysis in a defined structural arrangement suitable for the synthesis of the molecule PP via successive phosphoryl transfers, displaying kinase type activity. This process was extended to the phosphorylation of various substrates, in particular to the synthesis of ATP from ADP in mixed solvent and in aqueous solution in the presence of magnesium 2+ cation, probably via formation of a ternary catalytic species numbered as 8. Now, the latter abiotic ATP generating system has been coupled to sets of ATP consuming enzymes, resulting in the production of NADH by a combined artificial or let us say natural enzymatic processes. This is clearly shown to you here in this figure, where you can see the sequence of transformations catalyzed by the supramolecular ATP generating system. Now, you can see here 24N6O2, ACP molecule, the magnesium 2+ cations and the ADP, and the enzymes hexokinase HK, glucose 6-phosphate dehydrogenase G6PDH and 6-phosphogluconate dehydrogenase, that is 6PGDH. In the next figure, you will see the ternary catalytic species and the macrobicyclic thiazolium cyclophane. Templates possessing two hydrogen bonding subunits bind two substrates forming a ternary complex in which the substrates are positioned so as to facilitate bond formation between them. In a related manner, the rate and stereoselectivity of a bimolecular Diels-Alder reaction are substantially increased by binding both the diene and the dienophile within the cavity of a tris porphyrin macrocycle. Now, as you can see, I have discussed a macrobicyclic thiazolium cyclophane. This functions as a model of thiamine pyrophosphate dependent ligases and affects the benzoin condensations. Acyl transfer is catalyzed by formation of a ternary complex between a cyclophane receptor and two substrates. Difunctional binding and catalysis has been observed in functionalized cyclodextrines and in a hydrogen bonding cleft. Functionalized crown ethers have been used as reagents for peptide synthesis also. Let us come to the next topic, which is the discussion of bimolecular and abiotic catalysis. The design of supramolecular catalysis or catalysts may make use of biological materials and processes for tailoring appropriate recognition sites and achieving high rates and selectivities for reactions. Modified enzymes obtained by chemical mutation or by protein engineering represent biochemical approaches to artificial catalysis. This is also the case for the generation of catalytic proteins by induction of antibodies. Antibodies to reactive haptens are able to facilitate the transformation of the bound species. Generating antibodies against analogues of transition states should lead to a transition state stabilization and should facilitate this particular process. Such catalytic antibodies or abzymes have been produced for a variety of reactions, and an active field of research has been developed along such lines. It represents an approach to substrate specific, efficient and selective catalysis of supramolecular type, which is of much basic and applied interest too. The strong affinity for the transition state, that is TS of the reaction of a given substrate, leads it along the way and thus facilitates this process. In line with the remarks made at the beginning of this module, the antibodies should be generated not against a transition state itself, but against a TSA isoster lacking the groups X, which belongs to the reactive functions of the protein that is expected or that are expected to perform the reaction. This requires designing a TSA X species in which the phase presenting the X gap be ideally chosen so as to lead the induction in the antibody of the desired reactive functional group at the correct position. Now students, let us summarize this module. In this module, you have learnt that supramolecular reactivity and catalysis possess several properties such as substrate recognition, reaction within the same supramolecule, rate acceleration, inhibition by competitively bound species, and structural and chiral selectivity and catalytic turnover also. In particular, the transacylation reaction mentioned above, they operate on activated esters as substrates, but the hydrolysis of unactivated esters and especially the amides under the biological conditions, they present a challenge that chemistry has met in enzymes but not yet in abiotic supramolecular catalysis or catalyst. Development of supramolecular catalysts performing synthetic reactions that create new bonds rather than cleave them. By virtue of their multiple binding features, co-receptors open the way to design of cocatalysis for ligation, metallocatalysis and cofactor reactions, which act on two or more co-bound and spatially oriented substrates. Lastly, we also taught you that supramolecular catalysts are by nature abiotic chemical reagents that may perform the same overall processes as enzymes and without the detailed pathway by which the enzymes actually affect them or under conditions in which enzymes do not operate. Furthermore, and most significantly students, this chemistry may develop systems realizing processes that enzymes do not perform while displaying comparable high efficacies or efficiencies and selectivities also.